We are at the exciting interface where science meets industry. I'm going to highlight 4 main applications of a SNP chip that we have developed for tropical pines. SNPs are the most common type of genetic variation, which leads to biological variation between individual trees. These differences in DNA building blocks can ultimately influence a variety of traits, including growth, wood properties, and disease resistance. Our multi-species tropical pine SNP chip has the ability to detect the genetic variation at 50,000 carefully selected points on the 22 gigabase pine megagenome, uncovering genetic differences for important pine traits. For many years, breeders were dreaming about faster and more cost-effective breeding cycles. Commercially important traits such as wood quality are only expressed at maturity and can typically only be selected after 10 years in field trials. Our SNP chip will aid in fast-tracking this breeding cycle. Large numbers of informative SNPs can be incorporated in models to predict complex phenotypes via genomic prediction of breeding values at the seedling stage. The use of hybrids in pine breeding is becoming imminent. A good example is the susceptibility of Pinus patula, the most widely planted species in South Africa, to Fusarium succinatum. As a result, a number of pine hybrids have been produced over the last decade. Our SNP chip will be invaluable for pine species and hybrid identification. Forests have become more vulnerable to biotic and abiotic influences due to climate change. The SNP chip will enable analysis and prediction of genotype by site interactions in the form of landscape genomics. For example, we will be able to study the genotype by site interaction in areas with extreme temperatures, allowing us to make climate-specific selections in coming decades. We invite you to look at the rest of our publication for a glimpse behind the scenes on how we developed this tropical pine SNP chip.